Assalamualaikum and hi everyone. This video is recorded for the subject Reproductive Technology in Animals with the subject code ASH35603. We are from Group 2 and we will discuss about a journal article which talking about intracytoplasmic sperm injection. Intracytoplasmic sperm injection or ICSI is an assisted reproductive technique employed in humans as a method of choice in cases of male infertility. In livestock animals, ICSI has been used to study the interaction between oocytes and spermatozoa during fertilization as an important tool in the conservation of endangered species for the generation of high genetic value animals with the desired sex and as an alternative tool for the generation of transgenic animals. ICSI helps to avoid many of the steps normally necessary for the interaction between gametes during fertilization, such as acrosome reaction, sperm binding to the zona pellucida, and the penetration and fusion of the sperm with the oocyte plasma membrane. It was observed that in some species, sperm accessory structures such as the perinuclear theca, the acrosome, and cellular membranes interfere with sperm chromatin decondensation. The acrosome's content was found to be potentially harmful to the injected oocyte. Furthermore, the microinjection of oocyte during ICSI may result in alterations of intracellular ionic concentration, introducing foreign materials and preventing natural selection of sperm. ICSI can produce normal offspring in some animal species including humans, but injection of fresh and thawed spermatozoa also can cause obesity, aberrant growth, abnormal behavior, early aging, and increased incidence of tumor in the offspring of mice.
In natural mating, genetically imported but biologically inferior, such as low sperm production, poor sperm motility, and sperm abnormalities, males cannot be used. Like human, domestic animals including goats also can suffer from infertility or subfertility, which affects their lifetime production and reduce the number of offspring that may be acquired from a sire or them. Sperm with a low sperm count, abnormally shaped sperm, poor morphology, or sperm that do not move correctly, or motility are all causes of male infertility. Fertilization also fails when the previous IVF cycle had few or no fertilized eggs because the sperm cannot penetrate the outer layer. The egg outer layer may be thick or hard for sperm to penetrate. The best way to handle these problems is by ICSI, which can fertilize 50% to 80% of eggs. Although in vitro fertilization, IVF and ICSI are considered safe in humans, some studies show that they increase the likelihood of complication in the offspring. Several reports indicate that embryos of different species produced by ICSI had an increased incidence of chromosomal abnormalities, low developmental potential, abnormal patterns of calcium oscillation, and reduced cell numbers in some species.
It can achieve fertilization rates which can increase the number of fertilized eggs available for the transfer of raising. It can also learning how successful of ICSI works during IVF insemination. At the end, it will help the society for the biology of animal reproduction in the farm.
The objective for this study are to evaluate and compare embryo development, quality and gene expression in bovine embryos generated by ICSI and by IVF. The study included 3 experiments performed on bovine tumulus oocyte complexes (COCS). In the first experiment, oocytes from the same batch were measured in vitro and were randomly distributed to different treatments, which is ICSI, IVF, and SHAP, to assess in vitro embryonic development. A total of 592 oocytes were used for IVF, ICSI, and SHAP in the 9 replicates. In addition, pronuclear formation of ICSI's embryo was assessed 18 hours after insemination. In the second experiment, tunnel staining was carried out to examine embryo quality for the IVF and ICSI's embryo. In the third experiment, expression of selected gene was studied in the IVF and ICSI's embryo. Ovaries were collected from a local slaughterhouse. COCS were aspirated from 2 to 7 mm follicles using an 80 gauge needle. And then good quality oocytes surrounded by more than 6 compact layers of cumulus cells and a uniformly granulated cytoplasm were selected and measured in TCM 199 medium, supplemented with 10% of inactivated fetal bovine serum and 6 mg LH, 6 mg of FSH and 1 mg of estradiol, and then incubated for 18 to 24 hours. In vitro mature oocytes were randomly assigned to ICSI, IVF and sham groups. In vitro mature oocytes were co-incubated with separated frozen stock sperm per ml for 18 to 20 hours. In IVF TL medium supplemented with 0.2 ml of sodium pyruvate, 6 mg of fatty acid free BSA, 0.025 mg of gentamicin sulfate per ml and BHE, and 2 mg of heparin. Pisovantis zygote were stripped of human cell, veal, vortex and culture. Firstly, immediately before ICSI, 2 ml of sperm suspension was mixed with 8 ml of PBS containing 10% of whole vinyl pyrrolidone. Second, after 18 hours of cold culture, oocytes were denuded of granular cells by vortexing in the presence of 1 mg per ml of hyaluronidase, and then the mature oocytes were selected based on the presence of the first polar body. Third, ICSI was performed on an inverted microscope equipped with Hoffman optics. Normotype sperm with normal morphology were immobilized by tail scoring with the tip of an injection pipette before being aspirated into the direct injection pipette. An oocyte was positioned on the holding pipette with the polar body at 12 o'clock. After breaking the oiloma, the spermatozoa was injected into the oocyte. Sham injector was injected into the oocyte was performed in a similar manner as ICSI. Fifth, after ICSI, oocytes were washed 3 times in HECM, which is hamster embryo culture medium, HEPES, and then cultivated with 5 ml of ionomycin for 5 minutes, followed by incubation in K-salt, medium containing 10 mg of milliliter cycloheximide for 5 hours and culture. Next is we do the total number of cells, cell allocation, and tuning and L staining. A double staining was used to assess the total number of cells and their allocation to the true factotum, which is TE, and inner cell mass cell, which is ICM, in good quality expanded blastocysts generated by IVF and ICSI. 10 embryos per treatment were used. For the statistical analysis, differences among treatment were analyzed using one-way ANOVA after axis transformation of the proportional data.
To ensure the effectiveness of ICSI, the result is compared based on first, in vitro development and quality of embryos produced by IVF and ICSI, and apoptotic nuclei and gene expression in embryos. In vitro development and quality of embryos produced by IVF and ICSI, the cleavage rate is higher in IVF compared to ICSI, resulting a larger proportion of embryos generated by IVF, which is 36.1%, which the blastocyst stage compared with embryos generated by ICSI, which is 22.3%. IVF embryos generated a higher percentage of expanded blastocysts compared with ICSI embryos. However, no differences were observed in the total number of cells and trifactoderm cell between the expanded blastocysts produced by IVF and ICSI. Embryos generated by ICSI showed a higher number of ICM cells and the higher ratio of ICM cells to the total cell number compared with embryos produced by IVF. ICM, or also known as inner cell mass, helps in forming the embryo in proper way along with extra embryonic structures, apoptotic nuclei, and gene expression in embryos. There were no differences in the number of apoptotic cells between the IVF and ICSI blastocysts. Gene expression of insulin-like growth factor 2, which is IGF-2, in embryos generated by ICSI was lower than generated by IVF. Bovine embryos generated by ICSI and IVF reached the blastocyst stage by days 8 and 7 in culture respectively. This difference may result from a delayed decondensation of the sperm nucleus and the formation of male pronucleus in ICSI embryos. Delayed sperm nucleus decondensation or even failure to decondense may contribute to the low efficiency of this technique in cattle. Any alterations in the cell distribution between ICM and TE may affect the post-implantation development of embryos by the incidence of apoptosis of embryonic cells at the pre-implantational stage. Lower expression level of IGF-2 in ICSI produce blastocysts than in IVF produce blastocysts and causing abnormal expression of imprinted genes in SCNT embryos related to low efficiency of the cloning process. OCT4 is the earliest transcription factor expressed during embryo development and has a crucial role in regulating the initial developmental events. The higher expression level of this gene in ICSI produced blastocysts than in IVF produced blastocysts, which could be related to higher number of ICM cells recorded in ICSI embryos. Good quality embryo, which is functional mitochondrial activity and protection against ROS, are caused by SOD2 enzyme that protects cells against ROS.
In conclusion, this study showed that bovine blastocysts produced by ICSI insemination have a lower developmental potential compared with IVF-produced blastocysts. The decreased developmental potential observed in ICSI-produced embryos was most likely due to abnormal sperm nucleus decondensation that subsequently affected the fertilization and cleavage rate. In addition, the expression of genes important for normal embryonic development was affected by ICSI insemination. Overall, this result suggests that morphological and cellular characteristics of the embryos may not fully reflect their true developmental potential. It is expected that the use of ICSI will contribute to preserve the genetic diversity of endangered mammals, for which ICSI has shown good results. Currently, the ICSI technique is an unlimited source of information regarding the fertilization process. It offers a great potential for clarifying mechanistic differences among mammalian species with high impact perspective in both basics and applied research field. That's all from our groups. Thank you.